You have already learned about the relationship between the contractile component and the series elastic component in transmitting muscle tension to bone. But there are other anatomical relationships that contribute to muscle contraction, such as length-tension relationship. There is an optimal muscle length at which maximal tension can be achieved. This optimal length occurs before the onset of contraction, when thick filament crossbridges and thin filament crossbridge binding sites overlap. This is significant because optimal muscle length is coincident with maximal contraction strength. The percentage of maximal contraction strength that can be achieved decreases when the muscle fiber is longer or shorter than optimal length before contraction. Furthermore, because of restrictions imposed by skeletal attachments, muscles cannot vary beyond 30% of their optimal length in either direction. At the outer limits of this range, muscles still can achieve about 50% of their maximal contraction strength. The length-tension relationship also plays a crucial role in muscle receptor function. Muscle receptors provide the sensory information needed to control skeletal muscle activity and protect muscles from damage. These receptors are activated by muscle stretch, but monitor different types of stimuli. There are two known types of muscle receptors: muscle spindles, which monitor muscle length, and Golgi tendon organs, which detect changes in tension. A muscle spindle consists of a collection of specialized intrafusal fibers that lie within a connective tissue capsule parallel to the ordinary extrafusal skeletal muscle fibers. The muscle spindle is innervated by its own gamma motor neuron and is supplied by two types of afferent sensory terminals: the primary, annulospiral endings, and the secondary, flower spray endings, both of which are activated by stretch. A stretched muscle reflexively triggers its own contraction and resists any passive changes in muscle length. This occurs due to a monosynaptic pathway stimulating the stretch reflex and co-activation of alpha-gamma motor neurons. The stretch reflex pathway consists of 1. Afferent input from sensory endings of muscle spindle fiber, 2. Alpha motor neuron output to regular skeletal muscle fibers, 3. Gamma motor neuron output to contractile end portions of spindle fiber, 4. Descending pathways co-activating alpha and gamma motor neurons.
Tapping the patellar tendon with a rubber mallet stretches the muscle spindles in the quadriceps femoris muscle. The resultant monosynaptic stretch reflex results in contraction of this extensor muscle, causing the characteristic knee-jerk response.
Along with muscle spindles, Golgi tendon organs are another type of receptor that aids in muscle contraction. Golgi tendon organs reside in muscle tendons and detect and respond to changes in tension. This tension information is used subconsciously for smoothly executing motor activity. Unlike muscle spindles, the Golgi tendon organ allows you to consciously perceive muscle stretch or tension. If you have ever pulled a muscle before, you realize now how important the Golgi tendon organ is, and just how painful it could be if you do it again.